Let's we learn about microscope. What are magnifying devices? Magnifying devices are the instrument which is used to magnify the objects and see the object in enlarged forms. There are many magnifying devices. They are hand lenses, light microscope and electron microscopes. Telescopes also a kind of magnifying device. Hand lens is a very simplest form of magnifying device which is used to observe small soil organisms. Light microscopes are used to observe microorganisms such as bacteria and fungi, as well animals and plant tissues. Electron microscopes are highly technical and complex devices which is used to observe viruses. Let's move about magnifying devices. These magnifying devices are enlarging the smallest objects into large sizes. In these magnifying devices, there are lenses used to magnify the objects. Normally, convex lenses are used in all type of magnifying devices, but in hand lenses, there are only a single convex lens is used. Light compound microscope is a complex magnifying device with a number of lenses which is used to observe microscopic organisms, blood cells and plant and animal tissues. Electron microscopes are highly technical and complex devices which is used to observe viruses. Let's move about the history of microscopes. Many people experimented with making microscope. The first microscope was 6 feet long. The Greek and Roman used lenses to magnify objects over 1000 years ago. Microscope is invented in 1590s. The first compound microscope is invented by Hans and Zachariah Janssen of Holland. Anton van Leeuwenhoek and Robert Hooke made improvement by working on the lenses. Let's move what are compound light microscopes. Compound light microscopes are the complex devices which is used to observe microorganisms, plant and animal tissues and cells. As we know earlier, compound microscopes have several parts: eyepiece, body tube, coarse and fine adjustments, arm, stage and stage clip, strong base, mirror, iris diaphragm and condenser, objectives, revolving nose piece. Let's move the parts and functions of the light microscope. Base gives the support and stabilize the microscope. The light sources, they are in the microscope, provide light for viewing the slides. Diaphragm. Diaphragm controls the amount of light passing through the slides. Stage and stage clips. Stage is the place where the sample specimens hold in the microscope while we are working, and the stage clips are used to tighten the sample specimens without moving. Arms are useful when carrying the microscopes. There are objectives. 3 types of objectives found in light microscopes as low power, medium power and high power with a revolving nose piece. Eyepiece is the prominent part of the microscope. where we can see the object. Body tube connects nosepiece and eyepieces. Coarse and fine adjustments are used to move the objectives up and down. While we are using low power objective, we can use coarse focus, and in other two powers we can use fine focuses. How does a microscope work? There is an ocular lens in eyepiece and body tube objective lenses. When the sample object is kept on the stage of the microscope, then the light source gives the light and diaphragm controls the light to the stage. While we are keeping our eye on ocular lens, we can see the objects through low power first of all. After that, we can rotate the objective lenses into medium power and high power to get the clear magnifications. What are sample specimens? Sample specimens are the objects which is going to be under the microscopic observation. These sample specimens may be permanent or temporary. Permanent sample specimens are already prepared and cannot be removed. Temporary sample specimens can be prepared at the time of observation. Anything may be used as a sample, such as plant cell, plant tissues, animal cells, human cheek cell, blood cell, intestinal wall tissues. The picture illustrates the sample specimens what we can observe in our school laboratories. When we are learning about microscope, we have to learn about magnification. Let's move what is magnification. Magnification is the times of multiplication of ocular lenses and objective lenses, which means magnification gives the idea of how times an object is enlarging. This magnification of microscope is determined by multiplying the power of the ocular lens as well as the power of the objective lens. If it is observed under low power, then magnification of ocular lens is 10 and the magnification of low power lens is 4. So the whole magnification of the image is 40. Likewise, we can get the magnification for medium power as simple as ocular lens get magnification of 10, medium power lens gets magnification of 10, 10 into 10, 100 times of magnification. We can observe when we are seeing the object through high power magnification. As usual, the magnification of the eyepiece or ocular lens is 10 times and magnification of the high power objective is 40 times. So the magnification of the whole object is 400 times greater than the real object. Now let's move how to use a microscope. Place the microscope on a flat table facing to your body. Then make sure that the low power objectives small objective lens is in the position over the stage and no closer than 0.5 centimeters. Next, rotate the diaphragm to get your optimum light. After that, place your slide on the stage, adjusting it so that the specimen is directly under the lenses. The specimen should be in view when you are looking through the eyepiece. Then, focus on the specimen by slowly moving the coarse adjustment knob so that the slide is being moved away from the lens. After that, rotate the revolving nose piece so that the medium power objective is in position. Focus with the fine adjustment knob when you are moving the lenses up and down using medium power objective lenses or high power objective lenses. Rotate the revolving nose piece so that the high power objective is in position. Focus with the fine adjustment knob. Finally, when you are finished with the microscope, return to the lower power objective lens and remove the slide from the stage. This is the way how we have to use microscope while we are working. Now let's move. What about the observations? In the first diagram, you can see the blood cells. In the second diagram, you can see the cheek cells of human. Third diagram shows you the onion peels. Fourth and fifth diagrams shows some microorganisms, and the final observation shows you the plant tissue. Uses of compound light microscope. Here we go with how to use the compound microscope. A compound microscope is of great use in pathology labs so as to identify diseases. The uses of compound microscopes are large, vast. Normally in medical field and educational field in researchers, microscopes are used to identify samples, cells, tissues. Even in forensic experts can also find out the origin of a drug by viewing its component particles under a microscope. How to store the microscope after the use? Take off the slides from microscopes. Put lower power above the stage. After that, roll the nose piece up, stage down. Then turn off the light sources and wrap the cord around this microscope. Keep it in the wooden box. Save it safely. Proper storage of microscope will prevent or reduce problems. Optics and mechanisms of the microscopes must be protected from dust and dirt or funguses. While you are storing the microscope, you have to remember to cover the microscope with the protective covers, and you have to store them in low humidity environment. As well, remember children, always carry a microscope with one hand holding the arms and one hand under the base. Let's move about electron microscope. Electron microscope is used to observe the organisms very smaller than bacteria and fungus. Viruses are normally smaller than bacteria and funguses. These are observed under electron microscopes. Electron rays are used in electron microscope to observe viruses. But in compound light microscope, light rays are used as the light sources. Electron microscopes are used in medical field and pathology and medical research fields. Here you go with some questions, children. Related microscope, what we have discussed earlier, you will be able to answer all these questions. Who was the inventor of compound light microscope? Name some places where light microscope is used in. What is the proper way of carrying microscope from one place to another? Who was the founder of electron microscope? Where electron microscopes are used in? List out the parts of compound light microscope and write the functions of each part. The final question, if you are a scientist, what would be the object you want to observe first under microscope? I hope you will be answer these questions. Thank you, children. We'll meet with another lesson.